and in more depth about mercury and vernal pools. First of all, we would like to thank the land in which we could conduct our research, which is part of the Haudenosaunee territory, where we conducted a lot of our research. So this research was done majority last fall semester at Paul Smith College in the Adirondacks, where a group of Clarkson students, along with Evie Bromstead, who her PhD project is on this, and Professor Michael Twist helped out learning about mercury and vernal pools. What are vernal pools? Vernal pools are semi-permanent seasonal wetlands that don't have any fish in them, but they do have a lot of other important species and organisms that live and make it their home in these vernal pools. And they're shallow depressions that fill up with rainwater, mostly in the spring and the fall. They tend to dry up in the summertime, and in winter they freeze or there's just no water in them at all. So what makes vernal pools different from, say, wetlands is that they don't contain fish. They fill up with rainwater in the spring and fall months. They can dry up in the summer. Sometimes they'll be filled with water in the summer, depending on the weather, the weather patterns. And as I mentioned before, even though they don't contain fish, they're an important breeding ground for many species like woodland frogs, salamanders. So some plants and animal communities that you'll find in vernal pools are lots of deciduous trees. So when we did our research, we made sure we accounted for the different trees that were surrounding the vernal pool. So maple trees, oak trees, birch trees. There's also coniferous trees, so pine trees, fir trees, spruce trees. Wood frogs are known to live and breed in vernal pools, along with salamanders and fingernail clams, which is what we use to collect data off of to see how much mercury was in the vernal pools. So they were kind of our species or organism that we used in the lab to get mercury concentrations and things like that. So I'll turn it over to Hannah, who will talk about, is a vernal pool considered a wetland in New York State?
So are vernal pools wetlands? The definition of a vernal pool considers them to be seasonal depressional wetlands. However, the New York State definition of a wetland does not consider vernal pools to be wetlands like other states in the Northeast. According to New York State, a wetland is an area subject to periodic wetting and drying and is either at least 1 acre in size or adjacent to a body of water in which there is free interchange of water between the two areas. Therefore, this does not consider vernal pools to be wetlands, and they are not protected as such in the state of New York. Vernal pools do not get monitored or regulated like other wetlands that meet the requirements mentioned above. Other states, such as Vermont and Connecticut, do have regulations on vernal pools, just not something New York State has reached yet. Now, Anna can talk about mercury in vernal pools.
So first of all, what is mercury? Mercury is a unique metal that is considered a neurotoxin, and when it is exposed in high concentration can cause mercury poisoning. So a lot of the reason why mercury has been such a problem in the environment is due to human emissions of mercury that's caused pollution, that has caused mercury deposition to be prevalent in the environment, especially in New York State. We also, before I mentioned, we did most of the research in the Adirondack area, because those seem to be areas where mercury deposition is highest, and up along the St. Lawrence River Valley. So examples of where mercury can come from is coal-fired power plants. Emissions from western states can travel up with the westerly winds and come into New York State, and mercury gets deposited. And there's also things that have mercury, like household items such as batteries and light bulbs. In New York State, the Department of Conservation continues public outreach, regulation on mercury, and continues to monitor mercury within the state. This is because mercury can biomagnify and bioaccumulate up the food chain, which is detrimental to not only animals, but also humans, not necessarily with vernal pools, but again, in the environment. Mercury can get into freshwater systems, and if people are fishing and they eat that fish, that's where mercury can impose a threat to humans. So when mercury does enter vernal pools, mercury can become bioavailable due to organisms that are, or bacteria rather, that are methylating the mercury to then make it methylmercury. And methylmercury is mercury that is bioavailable for species to uptake within their system. And once animals ingest the mercury, it can bioaccumulate and biomagnify. And Hannah will go on to explain what biomagnification means.
So mercury biomagnification is something that occurs within vernal pools. Biomagnification is the accumulation of toxins in organisms that move up the food chain in greater concentrations. This diagram here shows how mercury can biomagnify up the food chain, starting in vernal pools with the decaying leaves, and can move up to larger species such as hawks. Biomagnification can provide data about the amount of mercury or other toxins in an area. In our case, that would be the Adirondack Park. It also can provide clues about disruptions to various ecological communities, and in some cases, as Anna said, pollutants can reach the human level and cause a human health risk.
And also to comment further on biomagnification, when we were conducting our research and we were doing background knowledge, the freshwater magnification, I thought this stat was pretty staggering. It has the ability to magnify up to 10 million times higher in higher trophic level species. So in other words, small plankton, for example, who uptake mercury, they don't get exposed to a lot of it or have high concentrations within them. But say a loon who eats the fish that ate the plankton, that then the plankton ate the other plankton, it accumulates and keeps accumulating, so that what a small amount of mercury for a zooplankton is 10 million times higher in a loon. And so that's where you can see how biomagnification and bioaccumulation of mercury is super detrimental to an ecosystem as a whole. I guess at this point, does anyone have any questions? Or if Hannah would like to add anything else.
I have a question.
Yeah.
So these vernal pools are these hot points in the wet landscape across this area of northern New York, from the St. Lawrence River up to the Adirondack Mountains. Can you repeat, if you mentioned it already, or just sort of emphasize the danger that mercury might have being mobilized in these what would seem to be kind of small areas? Or are there a lot of these enough that it would be a sort of by mass action quite an important player in mercury mobilization?
Well, first of all, vernal pools, as we studied in our research, we wanted to see if vernal pools have characteristics that make it easy for mercury to be easily methylated and mobilized. And we found that there are characteristics within also. So there's the Adirondack Park, which has characteristics such as pH levels and oxygen levels, tree cover, that all help and promote methylization of mercury. So then within that, there are vernal pools, and that we found some correlation with the characteristics of vernal pools that allow mercury to be methylated, which then for mercury to be methylated at higher amounts to then be input into the ecosystem. Hannah, do you have anything to say about the up the food chain, how vernal pools, because they are these small seasonal depressions in the woods, how can it be transferred outside of the ecosystem?
So it's known that the Adirondack Park is a mercury hotspot, based on studies done in lakes and rivers and the mercury levels in fish in that area. So we studied vernal pools and to see how that affects the ecosystem on a smaller scale, making them hot points within a larger hotspot. So we found that there was mercury in fingernail clams, and that can then be biomagnified up through the food chain into other ecosystems and impact other species outside of the vernal pools.
Great, thank you very much. There's a question in the chat if you want to answer from Sheldon.
Okay, I can't see. Hannah, could you read the question, please?
I'll try to find it.
Or, yeah, I can't see anything because my
Oh, there we are.
screen is sharing.
Okay. It says, this is from Sheldon. He says, just wondering if you have a map of your study sites as well. Might be outside the scope, but just wondering in your literature review or research how climate change may influence the biomagnification of mercury in vernal pools. So maybe you want to answer the question about where your study sites were located. And then the second question was, how might climate change influence mercury biomagnification in vernal pools?
So we actually had I wish we could bring it up, but we had a map. We took a GIS course last semester as well, Geographic Information Systems. If you're not familiar with that, it's where you can do a lot of things. So what we did is we took an outline of the Adirondack Park, and we were able to get GPS coordinates, and we recorded them when we went to each sample site. And we were able to plot those coordinates on a map using GIS. And so we created a map. I wish that we had it, or I wish that I could present it to you. We do have it though in our research paper. So I'm trying to remember where we sampled was so again, two major regions, the Adirondack Park and then up along the St. Lawrence River Valley. In the Adirondack Park, we sampled near Paul Smith's College, so the Vic Visitors Interpretive Center. Let's see if I can remember, even though it's only been a few months.
I have the map pulled up.
Oh, great.
So we sampled.
Do you want me to unshare, and do you want to share it?
No, yeah, I mean, we can do that if you would like.
Okay, I'm not good with Microsoft. Here we go. All right. So Hannah, if you want to share your screen.
It looks like we have one more question in the chat from Elaine.
Okay.
So it says, I have a vernal pool on my property in Ontario. Is there any way that I can protect its inhabitants from mercury?
Oh, good question. We will definitely answer that once Hannah finishes answering the other question. So Hannah, if you want to continue.
So we sampled 31 sites, half of which were in the upper St. Lawrence River Valley and the other half were in the Adirondack Park. The St. Lawrence River sites were up at Coles Creek and Wilson Hill Wildlife Management Center, and then in Potsdam on Clarkson's campus, and then 2 sites at St. Lawrence University. In the Adirondack Park, we studied sites at Paul Smith's and at the Vic, and then down in Newcomb at Huntington Forest, which is run by SUNY ESF.
And then to answer the other part of the question, climate change will definitely have an impact on methylation of mercury and therefore biomagnification and accumulation. One of the biggest issues is with the warming temperatures. With warming temperatures, there's an increase in the methylation of mercury to become methylmercury, which then is the bioavailable version of mercury to enter organisms and enter into the ecosystem. Things like rain patterns, precipitation, again, the warmer climate, temperature change, all has an impact. And from studies completed and ongoing efforts, all indicate that most likely there will be an increase in methylmercury to be bioavailable to then accumulate into the ecosystem. So definitely something that we have to watch and study closely, as the climate and the earth is always changing. Hannah, do you want to answer the other question about what we can do to protect vernal pools?
So protecting vernal pools, it's not a state regulated thing. However, you can do your part to protect your own vernal pools on your property, because it's a common thing on most properties that a lot of people have vernal pools and they might not know it. You can take steps to protecting them, just not by not allowing any destruction or harm to them, to keep the species and the ecosystem safe from any major changes. I don't know if you have anything else to add, Anna.
That I obviously agree with everything that Hannah has said. Said it very nicely. Also, just I think before I went into this project, I didn't know what a vernal pool was. So it's great that you were able to identify it in your backyard. And so, just respect it and make sure if you see any other vernal pools that, you're not it's harming it in any way. Now that you recognize that what a vernal pool is and how important they are to the ecosystem, and especially to salamanders and frogs, because that's their breeding ground in the springtime, just to protect it and not to try to minimize disturbing it. And easy things you could also do to prevent further, I guess, mercury contamination is make sure you dispose of your light bulbs and your batteries correctly, and because there's multiple household items that contain mercury, and if they're disposed improperly or you're not sure what to do with them, it can end up going into the environment. So if you have any questions about what contains mercury and how to dispose of household items that do contain mercury, the New York State Department of Conservation have lots of resources that are available to answer those types of questions, so that we can ensure a healthier environment and ecosystem and protect our wetlands and our vernal pools.
I have a question. Are there any kind of special plants or anything that can really thrive in vernal pools, that once they're there, they start popping up in them and such?
That's a good question. I'm not an expert on trees or anything. I don't know if Professor Twist, if you're still around, he knows a lot about plants and things. If you have anything to comment on that, or if you want to comment on that.
Yeah, that's a good question, Megan. What characterizes vernal pools compared to other types of wetlands is the animal community rather than the plant community. And so, vernal pools are fishless, which is kind of interesting. They're too shallow to keep fish in. But you can get mollusks like the fingernail clams they're talking about. And because vernal pools are so small, they tend to reflect the surrounding forest landscape. Now, whether or not the different animal communities influence the type of plants that are found within the vernal pool is a really interesting question, because we know that plants and animals interact with one another all the time. From just my limited experience with vernal pools, and I would venture that the students have more experience with vernal pools than I do at this stage after sampling 31 of them, is that we did sort of see a couple of different types, some that were dominated with sphagnum moss, and we saw those if we were in the St. Lawrence River wetland, or lowlands rather, or if we were up in the Adirondacks. There were some that had just beds and beds of moss in them, and they would stay moist that, and because they were moist, the moss was favored. And other vernal pools rather, were more like puddles, that were permanent in some cases, and again in the same sort of regions. Although last year it was relatively dry and very few were full of water. But in those ones that were not mossy, there tend to be a lot of leaf litter that accumulates, and so because of the leaf litter accumulating, they tend to go anoxic, so the oxygen concentration decreases. And because of that, you don't really see a lot of like emergent plants you might see in a wetland on the shore of a lake or a river. Also, because they're in the forest, the amount of light that shines down onto the wetland is very limited. It only sort of is there shining down when there are no leaves. And so they look a lot different than the surrounding landscape. That's all I can say. Good question.
Okay, thank you. Yeah. Yeah, that makes sense. And I believe one time, like for one of my classes, we also had to, I believe, go to like the forest, and I think we saw a vernal pool, I believe, and I think it was a lot of pitcher plants were like growing throughout there and stuff, and it was pretty cool.
Yeah. Pitcher plants often grow around sphagnum moss because of the acidity. And pitcher plants are they're the carnivorous plants that prefer those kind of environments. Yeah, that's interesting. Hannah and Anna, do you guys recall seeing pitcher plants in any of the vernal pools?
What do they? I apologize. I don't know. Like, what do they look like?
They look like a really thin wine glass, like a wine flute, and they have a hole in the leaf, and on the lip is curved. You often see them in bogs, growing on sphagnum in bogs. I don't recall seeing any pitcher plants in vernal pools, but I'm not saying that. Obviously, well, Megan's seen some.
Yeah, I don't recall. But I think that's also important to mention again, like the surrounding tree community. While we were doing our research, we made sure that we also wanted to see mercury content in the leaves within the vernal pool, so the leaf litter that would fall from the trees surrounding the vernal pools. And we accounted for like deciduous versus coniferous trees. And I forget, I don't know what was like, I can't recall if deciduous was more prominent than coniferous or vice versa, but we definitely accounted for that in our research. And as Professor Twist was saying, how vernal pools are more defined by their animal community rather than their plant community. Vernal pools are unique in the fact that these things called fingernail clams, before none of us, I don't think, knew what a fingernail clam was or like what they looked like and how small they were. And so it was pretty cool to be able to go out there and we literally would dig for these clams that were tiny. And it's just amazing to see what vernal pools, what kind of animals and organisms and species live in these things. So, whenever you're walking through the woods and you see like a vernal pool or even just a wetland, it's amazing to reflect upon how interconnected and complicated these ecosystems are and how vital they are to our environment as a whole. I don't know if Hannah has anything to comment about when we like our field work and what plants and animals we saw.
Our research did find that there was a correlation between percent canopy cover and the number of fingernail clams that we found at a site. So like I believe like sites with higher canopy cover had more fingernail clams that we found with mercury. Coniferous trees are more acidic, so we didn't get a chance to look at how pH levels in the pools affect mercury, but that's something that we're continuing to work on and we'll look at in the future. So, yeah.
Thank you for answering.
Looks like we have another question in the chat from Sheldon. Was there any correlation with pH in your study sites?
So as Hannah said previously, we didn't get a chance to look at pH, but that is something we're thinking about looking, like continuing to do and using pH in our future research. It's definitely obviously affects it. To what degree or how, we're not sure yet. At least in the areas that we studied and how vernal pools are concerned. But again, as Hannah said, we definitely think that percent canopy cover and the tree, like coniferous versus deciduous trees surrounding the vernal pool, definitely have an influence on the pH within the pools.
Yeah, Anna, you and Hannah can't see the comments, but I wrote in there that we have stored soil samples that all the students collected, and there's a method to determine the pH of the soil by taking some of the dried soil and putting it into a standard solution and using a pH meter. So that's actually one of the things that we will be doing, Sheldon, and it's a really good question because pH can influence the amount of methylation of mercury by bacteria. Thanks.
Okay, well, thanks for that, guys. Do we have any more questions from the audience? Okay, well, not seeing any questions, I would like to thank everybody who joined us for World Wetland Week and who came out today to hear Clarkson's presentation.
Yes, thank you for having us and allowing us to present. It's been a pleasure.
Okay, well, thanks, everybody. We look forward to seeing you next year for World Wetland Week again.
Thank you.
Thanks, everybody.
Thank you.